Welcome. My name is David Albertini. I am a scientist from way back studying basic aspects of reproductive physiology. And in 2014, I was fortunate enough to join the staff here at the Center for Human Reproduction in New York City and have been deeply involved in the research program here. It's over this period of 5-6 years now that we have come to realize that many of our basic understandings about how genetics, our inheritance of mothers and fathers' chromosomes at the time of fertilization, really needs to be revisited and reconsidered given discoveries not only by our group but many groups around the world. What I'm going to be sharing with you is somewhat of a surprising story, because as a scientist for many years we were taught that really the beginnings of development, when the egg and sperm cell come together in the process known as fertilization, reconstitute, from a genetic point of view, a new individual. That is, an individual that will receive one set of chromosomes, 23 chromosomes, from their father, which come within the sperm cell, delivered into the egg at the time of fertilization, and a set of chromosomes, 23, that will be inherited from the mother. And as we all know, it's the sperm that will carry either an X or a Y chromosome and be the primary determinant of whether that embryo will develop as a male or a female. In fact, just that fundamental understanding of the genetics of sex, if you will, is something that reproductive biologists and reproductive medicine specialists have known for a very, very long time. And it illustrates on one level the importance of the chromosomes that we receive, that we inherit from each of our parents. Now, for humans, compared to many other animals, we have known for many years that not all of the embryos that are made either during natural conception following intercourse or the embryos that are made here in vitro fertilization clinics to help patients have a family, not all of those embryos have the ability to implant in mother's uterus and develop for the 9 months that are needed for a live birth to have taken place. In fact, it has been recognized now, I would say, for a good 40 years, that when those pregnancies fail, often very early, often in the first 10 to 12 weeks of the pregnancy, also known as a miscarriage, when we examine the contents of that material, it was known some time ago that many of the failed pregnancies seem to have errors in the chromosome constitution of either the placenta or the fetal tissue itself. And this has led to the widespread belief that the success of a pregnancy in our species, in the human, really depended on the accurate transmission of those chromosomes, not only from our mother, when her ovary produces those eggs, but our fathers, as his testes produce the sperm that will end up fertilizing that egg, but even within the embryo itself. Which is to say that we always assumed that if there were chromosomal problems detected in the pregnancy, they must have come from an error when mom was making that egg cell. Maybe she made an extra copy or delivered an extra copy in an egg of chromosome number 21. And we knew even back then that if mother carried 2 copies of chromosome number 21 and the father delivered 1 copy of it, we would end up with 3, not 2, the normal number, copies of that chromosome. And in that case, children affected with Down syndrome would be born. So historically, I think it has been cemented into the minds of the general public as well as the professional societies that chromosomes were a single dominant element in determining whether a pregnancy would be established and maintained for the duration of 9 months. I must tell you, much to our surprise, we have now come to realize that it is far more complicated than that. And it is not simply a matter of chromosomes. It's a matter of the discoveries being made in the plasticity, the dynamic nature of not just the chromosomes, but the genes within those chromosomes. There now appears to be a remarkable degree of flexibility in how those chromosomes are apportioned into the cells of the early embryo. And even more striking, we're now coming to find out that while chromosomes were something we could see and measure, there are other aspects of our genomes, as we call it, the alphabet, the order, the encyclopedia of information that's embedded in the sequences of nucleotides of our genes, of our genomes, that there is a remarkable degree of rearrangement, modification, and sometimes elimination or accumulation of things called mutations, for example. Mutations that may not affect the development of the embryo or the fetus, but will give rise to individuals who are born with higher risk for disease later in life. One of the most common subjects being studied right now, where it appears that these very early changes in our genomes could be related to developing a disease later in life, has to do with autism and the autism spectrum disorder syndrome that we're seeing and diagnosing more and more in our society. So we would say, how is it that some individuals may be born with a higher risk of developing a disease like this later on in life, and others do not? It's a complicated story, but even though some of these errors and shuffling of genes is going on in that early embryo, and things that we can't detect by just measuring how many chromosomes or what chromosomes or structural rearrangements, okay, that many of those changes are either accepted into the genome of that individual or they're rejected. How are they rejected? It looks like these cells of the embryos are very smart. They can recognize some of these mutations, but not all of them. And in many cases, if it's a deleterious mutation, they can recognize it and fix it, or they can cut that piece of DNA out and eliminate it. So what does it mean to those of us who are taking care of patients? What does it mean for those of us who are interested in understanding the origins of human disease? And of course, in the particular case of the CHR, we look at this and we ask, what can we do to recognize differences between embryos that would be able to implant and produce a healthy child? After all, that's our single most important objective. And it's come back to testing for chromosomes, as has been pushed in this area as one of the add-ons in assisted reproductive technology, is really not very useful. It's a very crude evaluation of only a part of the embryo, so we're not seeing the global dimensions of the genome of this new individual that we have created by putting that egg and sperm together. And it's always been, I think, the sense of Dr. Gleicher and the Dr. Barad here at CHR that the maternal side, mother's uterus, the receiving side of setting up a successful pregnancy, should have more to say in what embryos are accepted and condoned during those critical early stages of a pregnancy, and what embryos would be rejected. And so in the end, we have come to understand that this is about much more than chromosomes. It's about much more than what some of the tests are that are being proposed to help us identify the healthiest embryo, or the embryo that is most likely to succeed during implantation and pregnancy. Rather, it's given us pause to take a look at other factors that do not have to do directly with the number of chromosomes. And this is where some of the brilliant advances, especially from some of the laboratories in Europe, have begun to explain for those of us professionals who are interested in the intricacies of this remarkable process of implantation. They have begun to explain what it is in the uterus, and the endometrium in particular, that needs to be established in order for the immune system of the mother to recognize, accept, tolerate is a word we use, the invading embryo, which itself is a very different genetic composition, of course, than the mother in which it's implanting. So we're looking now, I think, at a major shift in our thinking, and this has serious ramifications for conditions like recurrent pregnancy failure. But as I mentioned at the very beginning of this, many, in fact, the large majority of human conceptions do not produce a baby. Maybe 30%, maybe 35% of all conceptions ever will go to term. And this itself is beginning to explain how selective and how much discretion the mother must exert in deciding which embryos to accept into her uterus and which ones to reject. It is, after all, a huge, if you will, responsibility to not only allow for the establishment of the pregnancy, but to sustain it. A mother's physiology has to be completely adapted to the presence of the developing fetus, and it's a huge investment of metabolic energy, etc. So I'd just like to leave you with this very interesting time that we're at now in the field of reproductive medicine. We're still searching for the best ways to produce healthy embryos that will result in babies for our patients. But as time moves on, our thinking changes. So it's shifting more and more from chromosomes to the other dimensions of factors that may be secreted by the embryo, and, as I just mentioned, those vital signals in the endometrium, in the wall, in the lining of the uterus, that must be making a huge decision as to whether or not a mother is ready to adopt and sustain a pregnancy. So I'm going to leave you with that, and hope you'll continue to maintain your interest in our efforts here at CHR. Thank you very much.